Okay, well, good morning, everyone. Welcome to number 2 in the series of webinars that we're running from ProLab. For those of you that don't know me, which may be a few, my name is Mark, General Manager here at ProLab. We have 2 speakers for you today. Our own Charlotte Duncan, who's going to do the variant whack-a-mole. She's currently getting the props ready for that. And we also are very pleased to welcome Neil Bentley, OBE, who works over at COGS, who's going to fill us in on the work that's been done there. I don't have any intention of saying a great deal more other than to be here to facilitate and listen. So what I will do, if it's okay, I will just hand over initially to Neil, and we can start with the first talk. There is a chat function that you can use, and we will of course answer all questions at the end of each session, and if we need to stay on a little later afterwards, we will. I did notice on the invite that we didn't put a finishing time. We are aiming to finish around about sort of 10 to 12 o'clock, but if it runs on and people want to stay on and talk to us, then that's fine as well. Okay, thanks Neil.
Okay, right, I'll just try and share my screen, make sure I get this right. Right, can people see my screen now? Yeah, can people see the screen?
Yeah, we can see it, Neil.
Okay, right, thanks for that. Yeah, just want to check I wasn't talking to myself. Okay, so hi everyone, my name's Neil Bentley, and I'm part of Public Health England, and I've been seconded to help support COG within the PHE's COVID pathogen genomics program, and I form an interface between PHE and COG for the provision of the genomic service for probably the last 8 or 9 months. I need to state here and now that I'm not a genomics microbiologist. I'm just helping out, helping run the service, but I'm not an expert in genomics. So I'll leave the genetics questions to the experts. But just to make that clear at the start. So just on the first slide, I thought I'd better get the penguin in. As Mark told me I needed to get it in somewhere. It's in 2 places. You have to see whether you can see it in the next page. It's a bit like Find Wally. As you can see, as of today, the COG network has sequenced 392,487 genomes. And that's just the ones that have been uploaded into the CLIME database. There is a whole lot more than that either haven't been uploaded for a number of reasons, of which I'll go through a bit further into the talk. So I'll just see if I can change my screen. Okay, so just as an overview of what COG is. So COG's a number of university sites, 16 regional sequencing university sites with a central hub at the Sanger Centre, and you can see the 16 sites, and it covers in the United Kingdom, so Northern Ireland, Scotland, Wales, and England, and it's sequencing over the whole of the service, so not just England. And it includes the 4 public health agencies of Public Health England, Wales, Scotland, and Northern Ireland. It was initially put together, considered and put together in March last year, when Sharon Peacock realised that there was a need for sort of a coordinated genomics service across the UK. And some funding was made available from UKRI of £20 million to start sequencing, that ran until about September, October, and it was clear that the value of the sequencing, particularly when the variants came in, and people realised, or certainly the government realised, that these variants might have an impact. The amount of sequencing was asked to increase. The network was doing around about between 10 and 15,000 sequences a week, and they were asked to increase that to 30,000 sequences a week as of January, and some further funding was provided by NHS Test and Trace and the Department of Health and Social Care, and it was requested that that be administered through PHE, and that's where I came in September, and I'm looking after the £12.2 million fund of funds at the moment, and of course as of tomorrow we're getting some more funding to keep us going for a further 6 months while we transfer over to Public Health England. Okay, so the project administration is it's led by Sharon Peacock as the director. I don't think she needs any introductions. I think everybody knows her, and I provide a deputy director type role with on the side of COG. I'm not actually in COG. I'm PHE, but I help run the service. So this next slide. So what are the objectives of COG? And these were the initial objectives as they set them out. It was to generate data to detect mutations and variants, to combine with other data sets, including human genomes, so we can try and understand how the virus interacts with humans, to generate data to track the spread and support to the outbreaks within this country and globally for the pandemic, and as I've said, to support national, international collaboration. And a lot of the data is freely available via GISAID that anyone can use across the world. Okay, so the role, it's kind of morphed over the last 12 months. These are the main areas that it's associated with now. So it generates data for public health agencies, so they can have public health interventions. Its main function is to provide a research priorities, so you can actually understand the viral transmission, how it works, how it moves around, and we all know that some of the work that's been ongoing has made the government, led the government to make decisions about when we lock down and when we open up. So yeah, we take partial responsibility for identifying the variants just before Christmas and when we all had to go into lockdown. There's obviously the research on the mutations. I'll go into that a little bit more later. And also there's a big drive to look at the data and how we improve the sequencing tools and to look at the data and how we release the data and how we make that understandable for public health and clinical outcomes. The network, the COG network, also provides seminars and events, and there was a seminar last week, and I think there's another one in a few weeks' time. And obviously there's the national and international collaboration. So I'm just going to go into a little bit more about some of the studies that COG's been involved with, and you may well have been involved with some of these, it just explains, you know, some of the areas where COG is working, and it's so we can start to understand this disease. And I just say at the upset that it's really the first time we've been able to study a disease in such depth, in such numbers, because of the, you know, obviously the global nature of the disease. It's not like you know, Ebola in Africa, where you know there was some genomics undertaken in Africa, but it was on a much smaller scale, and we certainly didn't get the kind of information that we've got out of this. So here are the different studies that that we're undertaking. There's the HOKI study, which is the hospital onset of infection study, and it's to investigate how implementing real-time genomics can impact on the infection control to prevent spread within hospitals. And it's for patients and staff, you know, within the hospital setting, and they're looking at over 2,000 participants in over 15 hospitals. Professor Judy Brewer from UCL is leading the research, and early results show that rapid sequencing does enable identification of the source of hospital outbreaks in real time and fast enough to inform changes in infection control practice, and therefore further reduce the spread of the virus. I'm sure you're all pretty much, as being microbiologists, you all pretty much realise that would be the outcome. That, you know, as long as we can make it speedy, it can make a huge difference. The next study is the vaccination study, and that's simply to see can healthy people be protected with the vaccines, and do they evoke immune response? I don't need to go into the details of that, as we all know, and it's been widely publicised in the press, that the vaccines do work and they do evoke an immune response. Obviously, it's early days, and we don't know how long that response lasts for, but you know, the studies are ongoing there. There's the genomic study, which is a larger study than just SARS-CoV-2, and it's been running since 2016, when we were looking at you know, emerging infections within hospitals, for example, SARS, MERS, you know, flu variation, everything, and the study is to recruit sick people with sepsis and other critical illnesses, to try to understand their genomic picture by sequencing both the human genome in the patients and also the viral genome or the microbial genome, and linking it to the sequence, and plus bringing in all the extra metadata, you know, for example, date, you know, age, sex, postcode, ethnicity, etc., and bringing all that together to look genomically at the outcomes, and if there are any associations with the disease. The studies suggest that there is insight in how the patient and the virus genomically act together to influence patients' response to infection, and this will undoubtedly help with future outbreaks and with our understanding of why certain people are more prone to disease than others. So it's a really interesting study. There's the REACT study, where we're looking at real-time assessment of community transmission, and this is by using home testing techniques to improve our understanding of how the pandemic is progressing across England. It's the largest population study going at the moment, and it involves over 150,000 participants each month, and the findings provide the government with a better understanding of how the bug works and transmits, and the risks associated with the different population groups. You may well have participated in this if you've received a Ipsa Mori sample in the post, and to send back, so you may well have done that, have been part of that research. There's also the ONS study for the Office of National Statistics, looking at the protection survey, and it's to understand how many people of different age groups in the UK have COVID at any one time, and you'll see the difference between, you know, the government figures and the ONS studies, because as we know with this disease, A lot of people are symptomless with this virus. So it informs the status both within the NHS and possible disease trends. We're looking at both the antigen and the antibody in this study, and it will be running for a year. The participants within the study can visit either once, just as a one-off. They can visit monthly, or they can visit weekly to have both blood and swabs taken. So it will be really interesting to get the results of this, and I think the reports will be out pretty soon. There's also the SIREN study, and it's looking into the immunity and the reinfection rates across the whole of the UK, to see whether prior infection protects against any future infection of the virus, and what impact is detectable in any antibody, with the incidence of any antibody in the disease in healthcare workers. And initially, we found that over a period of 5 months, around 83% protection in this group against infection, over a 5-month period. Obviously, as we progress through the pandemic, we'll be able to see how this timeline extends. There's the Vivaldi study, which looks at how the virus is transmitting within the care home setting, with both care home staff and residents within care homes, and it's to inform future best practice. And there's 6,500 staff been screened, and over 5,000 residents across 100 care homes, and they're looking for both past infection, current infection, and also looking at antibody efficacies over the time, and the reinfection rates. So it's another important study. And as we move into opening the United Kingdom up, and also potentially as we move, we get future travel coming back in. There's a need to look at all travellers and importations, and at the moment we're screening all travellers coming to the UK, and looking for travel associated infections or any importations from other countries. And at the moment there's the test and release process where we're looking at both day 2 and day 8 screening in there, and any positives will obviously go on to be sequenced. And obviously speed is of the essence here. We can see this area increasing dramatically as we open up Benidorm, and as we all go off on holiday, hopefully this summer, but we'll have to wait and see what the government does there. And then there's just finally, there's all the data that's produced within CoV UK. It's uploaded through Climb into GISAID, which can then you can have free access in trusted research environments. So it's the IP of this data is it's out there for everybody to use. So I'll just carry on. So COG's been evolving. As of tomorrow, it's been around for a year, although the initial concept, the money was provided on the 1st of April last year, but it was evolving, mid-March. So it's had its 1-year birthday, and it was to, as I've said before, establish early proof of principle of the benefit of sequencing, for public health outcome, and then in November, as I've said, we realised the importance of the sequencing network, and it was expanded. And going forward, over the next 6 months, as of tomorrow, there's a transition plan to hand over the capabilities of the COG network, because it was obviously a research function rather than a clinical diagnostic service to the public health agencies of the UK. So that's ongoing now, and the funding that we've got is to provide that transformation. So just very quickly going through, just to check the time, just very quickly going through the operational structure. The samples come in through the Lighthouse laboratories and also the NHS. So pillar 1 through the NHS and pillar 2 through the Lighthouse laboratories. The Lighthouse lab samples go to the Sanger where they are sequenced, and the NHS ones go around to the other regional CoG sites. They're all uploaded into the CLIME database and then are made available to the public health agencies, where they're analysed, and then public health outcomes are understood, and then the government's informed as to how the virus is doing or mutating. And this is just a list of the NHS and PHE diagnostic labs that are associated with the CoG sites. You can see the different colouring schemes, and most sites, I think there's now about, this slide's slightly out of date, I think there's only 12 NHS and PHE sites in the UK that aren't feeding into the CoG network. So if you're in a lab that isn't, and you want to be involved with this, particularly after Professor Dame Sue Hill and Susan Hopkins sent out a letter last week asking for people to provide sequences. So if you are one of those labs, please get in touch. There's an email address there. And this is just showing the Sanger setup with the Lighthouse labs. So the shipments come in, and initially the Sanger was taking all plates from the Lighthouse lab, so that's both positive and negative samples, and then they were having to cherry pick those samples, store those samples in at -20, and then cherry pick the samples so they can then go on and process them through the NovaSeq process. It's now a little bit more easier now that only on the whole only cherry picked samples, so the Lighthouse labs have got cherry picking capability to just send the positive samples to the Sanger, which has made it a much simpler process. I mean, obviously at the height of the outbreak, none of us could cope with the amount of samples coming in, and there was only a proportion of samples sequenced. But at the moment, the Sanger has got a capacity of 25,000 sequences a day, and it is currently sequencing everything that comes in. There is a caveat saying anything with a CT value of below around about 30 doesn't generally sequence well. So the CT value of 30 is where we start to look for sequencing. And this just shows the geographical coverage across the UK. You can see that you know, on the whole, it's around between 8 and 10% across the whole of the UK. But at the moment, because the number of samples are lower, we're looking at as of the 1st of March, there was a 33% coverage in the UK, and some areas are doing much better than others. Wales, for example, have always had quite high levels of sequencing capacity in their region, and Scotland and the UK, and it's dependent on where the samples are coming in and how they're sequenced. So now just picking up on the variants of concern. As I said, I'm not a virology expert, and there's a lot of information out there on variants of concern, and I was just going to very quickly sort of explain what a mutation, a variant, and a strain is. I'm sure you already know this, but just to just to recap, a mutation is a single change in the genetic material of the virus. A variant is a whole sequence of the virus that may contain one or more mutations. And the strains with this SARS-CoV-2, we don't use the terminology because there is only the one strain at this time, and hopefully that will stay like that. So most viral mutations have very little impact on the disease and the spread. Some are actually detrimental to the virus, and they deselect, and some, however, some can make the virus more competitive. For example, increasing spread and the seriousness of disease. We haven't really seen any change in seriousness of the disease, thank Christ. But you know, we have seen you know, the problems with the increasing of spread and transmissibility. And there's a number of variants of concern. At the moment, I think I'll just bring up another slide, which I'll have. I can't share this slide because it's official sensitive, but at the moment there's currently 7 variances under monitoring investigation. There's a further 6 under investigation, and there's currently 1, 2, 3, 4 variants of concern that PHE are dealing with at the moment. But that's changing week on week. Okay, so this is just to say that if you weren't aware, they've changed the nomenclature of where the of how we understand about the variances, and made it easier to understand the variances. And I'll let you read through that, you know, if you get the presentation. So just in summary, we've the CoG networks stood up a wide, you know, capacity over the last 12 months, and we've gone from sort of 10,000 sequences a week up to 30,000 sequences a week at the moment, which can raise up to 70,000 sequences a week in early April, May time. We've currently sequenced around just under 400,000 genomes, and we're running at between 8 and 10% of all UK cases. We've developed the CLIME COVID database that allows us to analyse the data, and we've looked at you know, the importations into the UK. We've identified the variances of concern, and we've used the genomics, the information from the genomics outbreaks to change policy within the UK. There's been some quite good international recognition for the access that we provided, and we've had some pats on the back, particularly from the States, and we're collaborating both nationally and internationally, and the story continues. And here's just a number of the people who are involved with the CoG network, and just to say thanks to all of them. And you may be able to see it's not a ProLab penguin, but it is a penguin on the slide. So I'll stop now and open it up to any questions.
Okay, thank you very much, Neil. Fascinating. Clearly some great work going on there. A couple of questions did pop up. One of the questions was, would the presentations be available afterwards? They most certainly will. We've had permission from the presenters, so we'll get those sent out to everybody, and they'll also be placed on the website in the webinar section, so you can actually download a recording of this as well. A couple of hands popped up, which if everybody's happy, we can answer now. Diane, I think, do you want us to activate your mic so you can ask your question? Because I'm going to activate your mic. you there? Diane? No. Okay. Everyone's gone shy, but there is a chat function there, guys. So if you want to type any questions, are there any questions for Neil right now before we move on? I'm not rolling my eyes if you think I am. I'm actually looking up at different screens here, watching what's going on. Okay. Right, okay, well, pop them into the chat, guys, if there are any. So what we'll do then is, I think Charlie's now ready, and she's going to do whack-a-mole. Okay, Charlie?
Kerry, I think you need to start my camera. That's okay. Lovely. Hello, everybody. Let me just get my screen up. Okay. Does everybody see the presentation?
I can see it, yeah.
Super, thanks, Neil. Okay, so good morning, everybody. I think the first thing that I do want to say is thanks for everybody who's attended. I know it was a little short notice. I did personally want Mark to put this off until after the hairdressers opened, but you know, hey ho. So I'm going to talk a bit about testing for variants. There is going to be a slight overlap with a couple of the slides that Neil presented towards the end there, but hopefully it'll give a slightly different slant to the work that CoV UK are doing. So when we talk about testing for variants, asking to sort of come up with a title for a presentation, whack-a-mole seemed appropriate. It can seem very much like, you know, you're tackling one particular problem or one particular variant and another one pops up. There was news only, what was it, yesterday, the day before, about the double mutation that's arisen in India. So, you know, this is a very changeable situation, and that's hopefully what I'm going to, let's say, talk about a little bit from a commercial perspective. Some of the slides will be slightly out of date because, again, all of the variants of interest and variants of concern are constantly changing. So forgive me for that. Why do we have such attendance at this webinar? I think everybody is talking about variants, talking about mutant COVIDs. I always find it interesting when you look at the headlines of the different newspapers and the slant that they take on it. So, you know, and I think one of the interesting things from a personal perspective throughout the whole pandemic has been that, you know, more than ever, what we do in our job, our work, and what we've trained to do has been in the public domain like never before. I mean, I never thought I'd be asked by my parents what the difference between sensitivity and specificity is. Why are they having a swab test as opposed to a rapid lateral flow test? And I think as scientists and as you know, researchers and academics, I think it's quite challenging sometimes to distill, let's say, you know, decades of training that we've all had into a dinner table conversation. So, you know, that's just something that I've found quite interesting. And I'm not going to pass comment on the Prince Andrew headline. Perhaps he was just going to test his eyesight. So even more simple than what Neil was talking about, I find it quite useful sometimes to go really back to basics, because we can get so caught up in the science of it all. We all know DNA is made up of nucleotides. We have bases, and the bases form a pair. A always binds with T, T always binds with G. And when we're talking about mutations, there's three main types I think that we need to be mindful of. And the reason that they're relevant is because they tend to affect a codon, which is a sequence of 3 bases, which determines an amino acid. Subsequently, that amino acid combination determines the proteins that's formed. So when we're talking about these particular mutations, as Neil said, there are inherent errors, and oftentimes they're not particularly they don't confer any benefit to the virus. They're either null mutations or they're actively deselected because they don't do the virus any favors in terms of pathogenicity or infectivity. So the ones that do tend to be conserved and spread and become potentially future wild type, essentially wild type strains, you've got substitutions, deletions, and insertion. And you know, they each affect the codons in different ways. So a substitution is where a base is replaced with an alternative base, often to create a different amino acid. And then you have deletions and insertions, which, if you can see in the picture at the bottom right, you have a frame shift effect when you have those mutations. They're often the mutations that cause more negative effect, I'd say, on the virus, because you do end up with a whole shift of every single codon downstream or upstream of translation. So they're the three main mutations. When we're talking about why it's relevant, so this is the genome of SARS-CoV-2, and predominantly the mutations of interest that we're all talking about are around the spike protein. And the reason for that is because the spike protein, as we all know, is where the virus binds to the ACE2 receptor and infects host cells. Okay, when we're talking about mutations, it can get very number and letter heavy. And I think often people will, let's say, memorize these things because it makes you sound like you know what you're talking about. But in actual fact, the name and what's actually going on is not particularly relevant. It's the effect of the mutation that's important. So if we take a look at this list of mutations of interest, I think almost all of them listed there are in the spike protein, and pretty much all of them affect increased transmissibility. So if you note, there's really only the E484K mutation that affects, say, pathogenicity or vaccine effect. And if you think about it, that makes perfect sense. Viruses don't particularly want to kill their hosts. They want to spread and replicate. So when we're talking about mutations of interest, you know, there's two sides to it, really. Are we looking in terms of wider spread within the population, or are we looking at maybe reduced vaccine efficacy? And obviously the vaccine is our golden ticket out of all of this. So, you know, I think it's important that we keep an eye on that. The mutations that are listed here, there's a couple that, you know, I want to note perhaps and that haven't been listed. So the 6970 deletion, we haven't listed it because, I mean, in the UK, it's become pretty much the wild type strain essentially. So the Kent variant is our predominant variant that's circulating. It's interesting as well that this isn't the first time that the virus has taken a mutation such as that and become the predominant strain. So if you think about the original wild type that came out of Wuhan, that's not the variant that spread around Europe last year. We had the D614G mutation, which I think was found in Italy. Somebody will probably correct me if that's wrong, but that then became the predominant strain that spread globally. The mink variant, whilst it's been listed as a mutation of interest, if I understand correctly, the cull that was actioned in Jutland and in that area successfully removed that variant from circulation. But, you know, as we know, viruses tend to keep trying when they want to mutate in a certain way. So it wouldn't be surprising if this particular mutation appeared again. And one thing, again, you know, I'm personally looking at these mutations from a kit development perspective. You know, what's going to be the most useful things to look for? How are we going to formulate the kit? What's the application of it? What's the value of performing that particular test? And so if you look at one, for example, the Q498R, we will not consider including that in a commercial kit simply because it's a epistatic mutation. So you don't really see it without the N501Y mutation, and that is one of the main mutations of interest. So, you know, that's kind of some of the thought patterns that we have when we start looking at how we're going to be developing a kit to look for these types of things. So again, just a summary, as I said, I think I'm not sure if this is up to date, but this is available on the Gov UK website. We have access to this at the same time as all the rest of the public have access to it. So you can see that the nomenclature is very clear. You have variants under investigation and variants of concern. Okay, this headline, well, this news article made me laugh a couple of weeks ago or a week ago. Genotype assay testing is a new rapid novel technology. So basically what they're talking about is just a common SNP assay, essentially. So, you know, this is where, again, when the newspapers get hold of something, or when journalists get a hold of an idea, you can sit and look back and go, what are they talking about? You know, this is, how do you think we're currently testing for COVID? We're looking for certain regions of interest and we're detecting them using molecular methods. So yeah, it just made me laugh. This I thought I'd share. So just a couple of slides. And again, I don't want this to be, let's say, particularly a commercial. I don't want to talk about ProLab or ViaSure, but it's just to raise some of the challenges that I don't know how aware people would be. You have your own challenges in the laboratory to perform the testing. You know, you have the additional workload, you have all this new equipment to validate and bring on board. But on the other side, we have challenges in terms of supplying kits and reagents and solutions to meet your needs. So if we talk particularly about variant detection kits in general, so I'm not necessarily just talking about COVID, but any development of a variant kit, it's always a moving target because these things will always mutate. Mutations are random by their very definition, and combination of mutations can change the landscape. So again, if you consider the Kent variant, B117, then managed to acquire the E484K and became the Bristol variant. So you have, this is where the whack-a-mole comes up. You can say, okay, we have a well-characterized variant with the Kent, and then it acquires a new mutation and becomes something entirely different. Sequencing information and variant tracking is not matched globally. So what I mean by that is, you know, the work that CoG UK and the work that all of the collaborative laboratories have been doing has really put us ahead of the curve in terms of how we're going to manage variants and how we handle outbreaks and novel variants coming on board. That's not necessarily matched globally. So when you look for simple things such as, okay, if we develop a kit for the UK market, is it going to be appropriate for the rest of Europe? Well, how does that fit with South America? You know, what's the technology and the skills that can be applied? How do we best provide a solution without targeting too much one particular market or one particular area? Technology limitations, there's always going to be a trade-off in diagnostics. If we could provide something that is, you know, the highest quality data, let's say equivalent to sequencing data for, I don't know, a penny a test that you just squirt into a tube and press play and you can run 300, 400 at a time, then that would be the ideal. But there's always going to have to be some kind of a trade-off with quality of result, how much you're prepared to pay for it, and how much of the testing that you want to do. And I think, you know, talking to ViaSure users and talking to other people in the laboratory, one of the issues as we see it is that you're already pretty swamped doing your testing. And if variant testing is to come on board, first of all, is that necessary? What's the value of it? And then second of all, how can we make sure that when it is implemented, that it's as easy as possible for you to do whilst fitting in with your workload and work stream and, you know, things like all of your ISO accreditation and training and equipment maintenance and all those different things to consider. And then we have some particular challenges of developing a kit during a pandemic. And one of which, and this is not intended to be any kind of criticism, it's just an observation. And I fully appreciate the situation as it stands, but access to information as somebody who represents industry can often be very different to somebody who works for an NHS laboratory. And at the end of the day, we're all trying to, you know, work with the same problem and address the same challenges and provide solutions. So I think sometimes, you know, that I don't know if that's something that can change, and I don't know if it's something that can be improved, but certainly, I mean, Gov UK has a wealth of information on it. But again, we're finding out the information at the same time as the general public, as opposed to perhaps people who would work within the health service. The other thing as well that's quite interesting, and I don't think there's been a situation, at least not in my career, where I've had to deal with this, is the change of policy direction and guidelines. So you can have, I don't know, we're going to look for the RDRP gene, or we're going to look for the spike gene, because these are the ones that we're interested in. And that can very quickly change based on new information to say, in fact, you now cannot look for the spike gene. You must look for at least 2 other gene targets within a commercial assay. So this change is you know, it's appropriate, it's prudent, it's based on knowledge as we gain knowledge on the virus and as we get a better handle on how to address this whole situation. But it does leave us in a bit of a pickle when you consider that we have to go through re-registration of products, validation, and all of our own ISO accreditation as manufacturers and suppliers. And then a problem that we were talking about, I think, on one of the last BIVDA meetings, which is how do you, in a situation as a pandemic like this, where you have variants coming on board that people are going to need to start looking for, how exactly do you allow fair access to samples across all potential suppliers to do validation work? You know, and I know that that's something that DHSC are actively looking at, and you know, I think that that's necessary, and I think it's good. But I think it's also a consideration that I'm not sure how aware people, in the day-to-day lab running of a lab are aware that we could have this great idea, but in actual fact, how do we finalize it, finesse it, and then register it without access to these quite rare mutant strains, variants, sorry, and do all of the validation work. And again, this isn't intended to be an us against them, because I firmly believe that, you know, laboratory and academia and industry it's a symbiotic relationship. I think we all need to be working together, particularly at times of need in a pandemic, but there is some consideration that R&D takes resources, it takes time, it takes investment. And I think, you know, ownership of risk often falls on industry. That's the nature of the business. I'm not complaining about it, but I think it's just worth pointing out sometimes that, you know, it's not all rosy on this side. So a final just couple of slides, I'm conscious about the time, but just to say, in terms of variant kit options, we're still not completely sure about what the policy is going to be or what the steer is centrally, but we are ploughing ahead to create something that we hope might be a useful solution. We have an improved, let's say, COVID kit called the SUK2, which is the same as our current ViaSure COVID kit in that it has the ORF1AB and N genes of the Wuhan wild type. It also has the S gene with the 6970 deletion, so basically indication of the Kent variant. It makes it equivalent to what you would observe with the TAC-path S gene dropout. And then it has the endogenous control of RNase P, which I think is quite useful. And then we are developing a specific variant detection kit. So this is a multiplex single tube. Same format as all the other ViaSure kits, so it's going to be lyophilized, room temperature storage, runs from the same eluate as your first line COVID test, and it runs in exactly the same way as all the other ViaSure kits, so it's completely open to being put on any thermal cycler, and very simple prep, so it's 15 microliters of master mix, 5 microliters of extracted sample. We've decided to go for these 4 mutations of interest. The reason that we've done that is if you take a look at the analysis of the variants of concern and variants of interest, the route that we've decided to go down is to not say we have a kit for the Brazil variant or we have a kit for the South Africa variant, but rather we have a kit that detects key mutations, and based on interpretation, you can then essentially identify particular variants. So if you look at the result column, this is essentially the fluorophore channels that you'd get positive based on if you were to be testing one of these particular variants. So anything in green we will detect and also individually report out. So we can say this is the Kent variant, this is the South Africa variant, this is the Brazil P1 variant, etc. And then the ones in yellow is that we would detect them. So we would say that these particular mutations are here, but we can't differentiate between P2, the A23 and the B5125. So it's kind of a balance, but we figure that, you know, based on workload, on maintaining capacity and throughput by keeping it in a single tube multiplex, and by sort of really trying to focus in on what are we trying to achieve? Well, we're trying to look for the mutations that are going to be most problematic. And exactly what form they take and what variant name we give them, you know, that can change, but the key mutations are not particularly likely to change. Okay, and I think that's about it from me. So I'm going to stop sharing and hand back over to Mark.
Okay, thanks, Charlie. Fascinating stuff as always. I think everybody can see the chat. We have a question again. I'm not gazing into the starter, but I've got to look up there to read it. I think that was Jenny, wasn't it? Each pathology network is being supported by NHSC to stand up variant testing wave 1 rollout as early as next week. Neil, can you comment on that?
Let me unmute. Yeah, so the variant testing and the sequencing are obviously different, and I do know that there's a program to roll variant screening out across the NHS. For sequencing, I understand that that there is a move from the COG model into another model and as a service delivery model, and as far as I'm aware, and I know this is being mapped out at the moment, initially the PHE labs around the regions will be asked to provide sequencing capability for their region. As far as I'm aware, that's how it's going to go. But this hasn't been signed off as yet by the government, as far as I'm aware. But that looks like the direction of travel. In the future, will we have sequencing in every NHS lab in the country? I absolutely think so, because I think it's the way that the world's going, and you know, sequencing will become a mainstay test, because you can do it probably nearly as cheap as you can do an RT-PCR, and soon you'll be able to do it as quick.
Okay, what's the next one? Okay, so another question there: does the current viral kit miss detection of any of the current variants in circulation? Charlie.
Sorry, let me just come off mute. No, so under MHRA guidelines, we have to do in silico analysis of all new variants every 2 weeks. So we issue a report to all ViaSure users, basically assuring that any of the current variants in circulation are accurately detected by the existing COVID kit. So we provide full reports for all of that every 2 weeks.
Okay. I'm just looking down. I don't see any other questions coming up. There was, I think it was James that posted the note about us releasing the talks, but we will. Or another one just came up. So the talks will be released, and they'll also be on the website. So question there, comment from Jennifer.
Jennifer, would you like to come on and speak to that, because I hadn't heard this before.
Yeah.
But it sounds
Jenny,
quite
do you mind coming on camera or chat? Okay, I think we can hear you now, Jennifer. You there? Oh, there she is. Right. Okay. Are we having a technical hitch?
I think you've been unmuted. No. Oh, the joys.
Oh. Okay. We've pressed every button we can find.
I mean, has anybody else on the call been involved in this first wave rollout trial thing?
Sorry, Jennifer.
It's
kind of put you on the spot here, I think.
I understand Mel Smith has been heavily involved with this rollout. I don't know whether Mel's online.
Don't know.
Yeah.
David Wells.
Yeah. Okay. Yeah. So each network has been asked for a volunteer. Okay.
Yeah.
Okay. What we'd like to do actually is maybe for Hertie, for Rachel, and for Jennifer, we can have a chat afterwards. We'll get in touch. Okay, another question's come to how do we conduct validation in low prevalence? Ooh, well, yes, that's that has certainly been a challenge for us, and we are very much engaged with the MHRA about that, because the document was written. I think when Charlie said before that we don't get a lot of information at the same time that the labs do, we tend to get it at the same time it goes into the public domain. And that tends to happen is that when they're putting together protocols about validation verification in labs, they write these things in silos and send them out and then consult us to see what we think. And we usually find that it creates problems. Charlie's grinning from ear to ear though, so I know she wants to say something about this.
No, honestly, it's a challenge. It's a challenge, I think, whether you're developing a new kit or whether you're trying to implement a new kit. I think on the flip side as well, you know, that there was challenges during the peak of the pandemic when you had high prevalence. I think whichever way you slice it, there's going to be difficulties. I know that we did a webinar a couple of weeks back on the Neo Biobanker. Now that's opened our eyes to a whole new world of biobanking and things. And I know that there's a consortium within the UK, and I forget the acronym, TCDC, something like that. And I know that they're sort of primed and available to help with sourcing samples for that purpose, for validation and verification work. But no, I don't envy you on having to do it. We have our own challenges on trying to validate new kits. So, yeah.
Okay. Right. Okay. So there's a few comments coming up there, but I think we are covering them. But certainly, Hergy, Rachel, Jennifer, we'd like to have a chat if that's okay, and we can maybe then put something together as a tagline note to this. All that remains really, I think, if there are no further questions, is for me to thank Charlie, who's only in the room next door, and Neil. Thank you very much, Neil, for your support and contribution once again. Third webinar in this series will roll out probably in about 3 to 4 weeks, give us all a bit of a breather after Easter, and there will be a lot more information available then about the variant. And then we're moving, a little bit of a teaser for you, the next ones after that are actually going down the sepsis route. We fancy a bit of a change, just a new venture that's coming out for ProLab on point of care. And looking down at these notes here, and I don't think anything else has popped up there. No. Okay, so thank you very much again, all of you, for attending. I know you're very busy. I know Wednesdays are busy for the virology meeting that we did clash with. There was actually about 30 or 40 people that wanted to attend that asked for recordings as well, which we will send out. And keep up the good work. As I said at the end of the last one, I do think we're the hidden health service. Nobody stands outside houses and bangs pans together for us on a Thursday night. Maybe they should. But keep up the great work, guys. Stay safe and stay sane. Okay, thank you. Bye-bye.